Hello, I'm Stephen Lee and welcome to Omics Quest. Today's video is the first in a series called Inside 10x Genomics, where I'll be breaking down key concepts behind 10x Genomics technologies. As many of you know, 10x has been a leading pioneer in the field of single-cell technologies, and I truly think what they have developed is incredibly cool. So be sure to stay tuned for future videos as well. Today, I'll be diving into how single-cell 3' gene expression libraries are made. Let's break it down. This video assumes you have some basic understanding of 10x single-cell technologies. Briefly, you start with gel beads, use microfluidics to create gel beads in emulsion or gems, synthesize cDNA inside each gem, then pull the gems to amplify the cDNA, and generate sequencing-ready libraries. Let's take a closer look at what happens after a gem is created. Inside a gem, there is a gel bead and mRNA molecules released from a cell or nucleus after lysis. To keep things simple, let's focus on the cDNA synthesis of a single mRNA molecule. Here's the sequence structure found on a gel bead. The mRNA molecule of interest binds to the poly-T sequence like this. From here on, reverse transcription takes over using a template switch oligo or TSO to generate the first full-length cDNA strand. I won't go into the details here because I already have another Omics Quest video called Quick Guide to cDNA Library Construction and Template Switching Oligos, as shown here. Check out that video if you're interested in learning more about how template switching works. At this point, I want to emphasize that all of this is still occurring within individual gems. Once the first strand of cDNA synthesis is complete, the gems are broken and the cDNA strands are pulled together for further processing. After the cDNA strands from individual gems are pulled together, they undergo PCR amplification to generate sufficient material for library construction, as shown here. Once cDNA is amplified, it undergoes enzymatic fragmentation and size selection to optimize the amplicon size, 300 to 400 base pairs, as full-length cDNA is too long for next-generation sequencing. Next, the Illumina read 2' primer sequence is introduced. Finally, a second round of PCR is performed to introduce sample indices along with Illumina adapters. This is the end result. You can see that even though we started with full-length cDNA, only the 3' end remains in the final library. This is why the assay is called single-cell 3' gene expression libraries. But this is totally fine for quantifying gene expression, i.e. determining how many transcripts of each gene are present in each cell. By counting reads that map to the 3' end of each transcript, we can infer the expression levels of the associated genes. Nice! Thanks for watching Omics Quest. If you enjoyed this video, please like and subscribe to the channel.